Yeah, hello girls. Last time we started with dihybrid crosses, and you will remember that dihybrid means that we are looking at the inheritance of two genes together. So we are going to look at an example of guinea pigs, and the two alleles we are looking at is whether their hair is short or long, and the color, which can be black or white. So here are our two guinea pigs, and remember the terminology. The P generation is a parental generation, and the way these crosses are set up, we usually start with two homozygous individuals. So in this case, this individual is homozygous dominant for both traits. So being black is the dominant trait and being white is the recessive trait. And this individual is also long-haired, so the other gene or long is dominant for the other gene, and short hair is recessive. So this one showing the dominant phenotype and this one showing the recessive phenotype. Now in the next of, because they are homozygous, they can only make one type of gametes, and the gametes carry one copy of each of these alleles. So they carry big B, big L, and here same thing. They also can make only one type of gametes, little b and little l. Now during fertilization these gametes will fuse, and as a result you get offspring, and each of their offspring will be heterozygous, having inherited the dominant alleles from one parent and inherited the recessive alleles from the other parent. So in our so-called F1 generation, all the offspring is heterozygous, and because we are looking at an inheritance pattern with complete dominance, where the dominant allele masks the recessive allele, both of these individuals show the dominant phenotype. So they're both black and they both have long hair. In our next generation, we are now crossing the two heterozygous individuals, and they can produce four types of gametes. These gametes can either carry the dominant allele for both genes, one dominant and one recessive, the other recessive and the other dominant, or two recessive. So four possible gametes. So we need to set up a Punnett square with four possible gametes on each side, and the Punnett square has 16 fields. When we do this cross, then this is what we're going to get. So the majority of them will be black and four of them will be white. So when you have a closer look, you see that all of the black ones carry at least one dominant allele for color. So they all carry a capital B, and they're either homozygous or heterozygous, but doesn't matter as long as they carry one, because this is complete inheritance, complete dominance. At least as soon as they carry at least one of the big B's, they will show the black phenotype, whereas all of the white ones are homozygous recessive for hair color, and therefore they are white. Now let's have a look at coat length. You see that all of these one here, this one and these three all have long hair, which means that they carry at least one big L, and this one, that one, that one, and that one are homozygous recessive for coat length, so they have two little l's and therefore they have short coat length. Now when you combine these two, you find that 1, 2, 3, 4, 5, 6, 7, 8, 9 both have black hair and long hair. 1, 2, 3 are black with short hair. 1, 2, 3 are white with long hair, and 1 is white with short hair. So in this cross, in a dihybrid cross, your typical phenotype ratio is 9 to 3 to 3 to 1. Now remember, this is only a prediction. If you actually cross two animals, you will never get the perfect ratio. Only if you looked at hundreds and hundreds of guinea pigs, then it would eventually look like a perfect 9 to 3 to 3 ratio. But if you only look at a small number, the actual ratio can always deviate a little bit from the perfect ratio that you would expect by doing these Punnett squares. I'm saying that because often in a question they will give you actual numbers of a genetic cross, and then they will ask you, now what kind of ratio does that resemble, and why is the actual ratio not looking like the perfect ratio that we would expect. And then you have to say that, well, when you have a small number of individuals, it can deviate a little bit from the perfect expected ratio that you get when you do a Punnett square. So as we just counted, 9 of the individuals will be black with long hair because they carry either 1 or 2 dominant alleles for both of the genes. 3 of the individuals will be dominant for color but recessive for for hair length. 3 of them will be recessive for color but dominant for hair length, and then 1 will be recessive for both genes. And this is the typical ratio that you are expecting for this type of cross. Now let's go back to this one. This is easy to count when you write these genotypes in the matter that we've discussed, in the way that we've discussed earlier. So you need to decide which letter comes first. Do you want to have the B first or the L first? In this case they chose to have the B first. And then you always write the alleles that belong to one gene together, and you always write the capital letter first. And when you do it that way, you always end up with this type of pattern. So 4, 5, 6, 7, 8, 9. These are your nines. 3, 3, 1. If you set it up in a different way, you still get the same ratio, but it makes it a little bit more confusing and harder to count the Punnett square. So you make your life easier for yourself if you follow this rule of setting up your dihybrid cross. And that's it for today.